Hi, welcome. Today I'm going to be talking about this protein called Isthmin1 and its potential to extend human health span. Maybe or maybe not, as I will discuss later. So today's presentation is formatted in a way that's a partially journal club and it's partially presenting my research. And this is a paper that I'm going to review. So it talks about the Isthmin1 being a novel biomarker of aging in killifish, a type of killifish, which was published around in 2022, which is very recent and was done by Ocean University of China. And this is the moral of the story. So the small molecule or chemical called salidroside, which can be found in the extract of this flower called Rhodiola rosea, may have therapeutic effect against aging and promote longevity. And here it's in Korean, it's saying that this is the flower, the Rhodiola rosea, maybe the flower of the plant that the Emperor C was looking for back in the time of ancient China. And this is summary. So Isthmin1 protein is an adipokine, so it's a hormone. And obviously, because it's a hormone, it's highly present in these endocrine tissues, but especially in adipose and thyroid tissue. And it contributes to the general well-being of organisms as it combats against cancer as well as virus, which is good. And it also controls blood sugar level as found really recently because it has a similar function to insulin, which is very interesting. And what the authors are claiming in this paper that the increased Isthmin1 level actually extends the lifespan and killifish, and reduction of the protein is actually correlated with aging, and you can actually promote Isthmin1 level by salidroside, is what they're saying. And so the salidroside has a potent rejuvenating potential. So this is the general description of Isthmin1 gene or protein. So here is the gene. The Isthmin1 is located in the chromosome 20 in humans, and it has around 77 to 79 kilobases long of bases. And it has 6 axons. So as for the protein on the N terminus, it has the signal peptide because it's a hormone, and has a TSP and really highly conserved AMOP domain. So over here, showing the killifish and human Isthmin1 protein as 3D structure, and they look alike because they're highly homologous. On the right side is the as a figure to show that Isthmin1 has multifaceted function, as I discussed or introduced in the summer slide, and also has the metabolic activity, as I told you. So this is the first result they've shown, and Isthmin1 level reduces with age in killifish and human. And what the authors on the left show is that killifish across many tissues has decreased Isthmin1 and when the fishes age, and they provide an additional evidence in human blood that a 30-year-old human has higher expression of Isthmin1 than a 70-year-old human. So Isthmin1 level decreases with age. And what they also showed was that Isthmin1 is a potent antioxidant. Over here, they've shown the control and the experimental group, and the experimental group was actually treated with the R Isthmin1, which is recombinant Isthmin1. So the supplement of Isthmin1 for higher expression actually reduces ROS in the same period of time. So ROS level reduced, which is followed by protein oxidation reducing and lipid peroxidation levels reducing. So treating this killifish is actually lowered ROS level, which follows the ROS theory of aging. And salidroside is what you're claiming to be having effect on age through Isthmin1. Salidroside, otherwise called SDS, extends lifespan in killifish. So on the left side is the muscle and liver tissues from the killifish, and the control group were compared against the SDS group, which is treated with salidroside, and they've shown that there is a general increased expression of Isthmin1, which means that the aging the decrease in Isthmin1 level by aging was actually delayed by salidroside. On the right side, they showed that this was actually correlated with the increased survivorship. So around 5 weeks more or so, the killifish lived more. So that was their results by Lee et al. 2022, but our results unfortunately is contradicting with their results, especially about the humans. So when we actually tested in whole blood of the humans, the Isthmin levels actually increased with age. So on the right side over here is showing our expression distribution of 1,130 samples that we have, and we can see that this y-axis is expression and x-axis is the age group. There is an increasing trend, but from teenagers to 70-year-old elderlies. But for some reason, 80s and 90s have lower level of Isthmin1, but this may be due to the lower sample size, or maybe because the effect size of change is not that much. But the important thing is that what we found in our prediction model, so on the left side of here is our aging prediction model using the RNA or gene expression. So on the y-axis of this expression model presents the list of genes that are important for contribution in age. And we found that over here, the absolute contribution score, the Isthmin1 is second highest. So Isthmin1 obviously is important for aging according to our clock. But interestingly enough, so the genes over here are masked or blanked for obvious proprietary reasons. Over here on this plot, if you have the more pink or showing the more expression on the right side of the plot, so positive contribution, which means that the Isthmin1 level, the higher expression of Isthmin1 level is promoting aging, so it accelerates aging in our model. So that really contradicts with the claim by Lee et al. So whole blood level is actually, in the whole blood of humans is actually, we claim that it has negative effects on aging. And so we've actually wanted to validate this in other studies as well, other data sets as well. So we've used a public data set provided by GTIX, and which obviously has more tissues as well. And we found that Isthmin1 level actually generally increases in other human tissues as well. So here we've chosen the adipose, not adipose, but endocrine tissues, and the other tissues that the Lee et al. Showed, and we showed that that the adipose tissue, breast tissue, and liver tissue all has this increasing trend, general increasing trend of Isthmin1 level, and slightly, but not significantly in whole blood, apart from thyroid actually. So maybe the effect is coming from the thyroid, I don't know. So yeah. So in conclusion, Lee et al. Claims that Isthmin1 reduces with age, and its supplement has anti-aging effect via ROS reduction. However, our data suggests that Isthmin1 conversely increases with age, and even our model even predicts it to have an accelerating effect in aging, which is contradicting to the claim, unfortunately, again. So the effect of Isthmin1 is still dubious and should be doubly checked by other research. I am really wondering what it looks like in other human tissues and even mouths as well.